Hi guys. Hopefully we're live. I had a few technical difficulties setting up, but I think it's working. So give me a quick thumbs up or a hello or something if you can see a PowerPoint, and ideally see me as well, although the PowerPoint's probably more important. And if you can hear me. If you can do all three of those, then we're good to go. And obviously, if the sound's not working, you're not going to know what I'm saying, because I'm a little bit concerned. It's making a slightly weird noise in my headphones. So I'm hoping it's working. I'm getting an error message on my one. Oh, okay. No, I think it's working now. Yeah, can you guys see and hear me? Okay. I'm going to share this PowerPoint. Okay. Okay, now you can hopefully see a PowerPoint. I don't know if it was working. It seems to like be connecting and disconnecting. Yeah, I'm going to make a start, and I'm hoping it's working properly now. So yeah, welcome to a Snap Provides Sunday session. If this is your first class, yeah, these sessions are just to introduce you guys to our tutoring programs. So we run classes like this 4 times a week in biology, chemistry, physics, and maths on our website. You have to subscribe to a package called the Ultimate Package, and yeah, it's like a really helpful way to boost your biology knowledge, and obviously also the other subjects. Oh, Zena, I have seen your things now. They just popped up. So thanks for confirming that. Cool. Where's everybody joining from today? Have we got an international crowd in today's session? And yeah, can you guys hear like a staticky sound? In my headphones, I'm getting this static noise, which if it's just affecting me, that's totally fine. If it's also playing to you guys, I'm sorry about that. I don't know what it's from. And yeah, well, hopefully you guys can't hear it. So yeah, we are doing carbohydrates today. This is one of the biological molecules topics. So hopefully you guys will have done this with school already, even if you're like doing AS. Yeah, most schools will have done this in the first half term. If you haven't done it, no problem. It should still be possible to keep up with what we're doing. Yeah, so as usual, we'll start with our sort of learning pyramid. These questions that get us in the sort of yeah, the idea of the topic for today. So yeah, first question. Can you guys tell me what is the scientific name for sugars? And I'm getting a thing saying I can't connect to chat, but I don't know why. Okay, your message is coming through now. That's much better. Oh, it just disconnects straight away again. I can see your last answers: Essex, Dakar, Sri Lanka, Harlow, Karachi. Oh yeah, so we're all from all over. I'm actually doing this lesson today from Morocco, so adding another country into the mix. But yeah, I need to try and get this chat working. Why would it not work? I know. I'll join the YouTube session from my phone. Then I'll be able to see it. Sorry for the delay here. Because yeah, my laptop, like, okay, I'll give it one more go. No, yeah, when I refresh the page, it shows the messages, and then as soon as and then just doesn't do it again. I don't have to keep refreshing it the whole time. So yeah, this will be worth the 2 minutes that it'll take to set up. This is such an ordeal. I've got three devices now connected to YouTube to do this. So how do I see the chat here? I've got to be able to make this work somehow. Can you chat on the phone? Are any of you guys joining from a phone out of interest? And can you see the chat? I'll have to refresh this page to see your answer. Supervise live streams. Comments. I can't see a way to view the comments on my phone. And this is probably just me showing my age and technical ineptitude. But yeah, okay, that plan then is not going to work, sadly. I guess what I'll have to do is I'll have to ask you guys questions, and I'll have to refresh my page every time to get your answers. It's certainly not ideal, but we'll make it work for today. And yeah, apologies. I don't know why it's not working. You can see chat on the phone. Okay, I just looked for a little while and I couldn't find it. So we'll use this makeshift method for now. But yeah, try and get something, obviously try and get something better set up for another time, because this is a tad ridiculous. But you guys are already here now, so I don't want to keep you waiting while I try and get it working better, and this will do. So as you guys have all said, saccharides is the technical name for sugar. Cool. Then yeah, what is the name for lots of sugars linked together? Yeah, nice. Polysaccharides. Perfect. Nice, Christopher. First to answer that. And what type of organism can produce sugars? I guess let's make it more specific. From inorganic molecules. Because basically anything can create sugars from other molecules. So humans, we can make glucose from, say, like an amino acid, but we can't make glucose from carbon dioxide. So we can't turn inorganic carbon into organic carbon. That is the job of plants, as you guys have said. Yeah. Do you have a more technical word you could maybe use instead of plants? Possibly if you're AS, you won't have come across this, but any A2 students joining, or actually to be fair, I think you should all know this from GCSE. There's another word, two other words, in fact, that you could use here that would be actually slightly better to put in an answer. Flora is a good guess. Flora is like a technical scientific name for plants. So yeah, I like your thought process. But yeah, I wasn't actually thinking of flora. So this isn't a synonym for plants. This is a synonym for something that makes sugars from inorganic molecules. Otherwise, flora would be a good answer. Yeah, so I was looking for either producer or autotroph. So yeah, like I said, if you're A2, that should be familiar. If you're AS, you might not cover that yet. Okay, nice. Who can give me the balance symbol equation for photosynthesis? So I know this is a little bit awkward to type. So don't worry about doing this, like the subscript letters. You can just write like CO2. Yeah. And even then it's a little bit awkward to type an equation anyway. But yeah, hopefully this is one that you guys should all know. Some people will learn it for GCSE. And if you did, oh, nice. I can see your guys' answers now. Producers, autotrophs. Yeah, nice. Perfect. I'm kind of having to work. I'm going to always be like 15 seconds behind you guys because I can only press refresh so often. I'm doing it as often as I can. But yeah, I might not get every answer straight away in real time. There's a, what's the shortcut for refreshing a page? Is it like an F key that you can press? I wonder if that will help. I just press refresh. Sweet. Yeah, nice, perfect. 6H2O plus 6CO2 goes to 6O2 plus glucose, which is C6H12O6. So that equation should be super familiar to all of you, hopefully. If you haven't memorized it yet, just memorize it. The beauty of this equation is you get two equations for the price of one, effectively. Because if you just reverse this equation, then that's the equation for aerobic respiration. So yeah, and that's another one you need to know for A-level. Cool. So then what is the process by which sugar is used by living organisms? What would you say for that? Yeah, nice. Cellular respiration. Or just respiration. If you put aerobic, that's great too. You actually don't need to, for this question, you don't need to put aerobic because it's not specifically aerobic. But one thing that often lets people down is when you do actually need to put aerobic respiration, people just put respiration and you don't get the mark. So say there was a question like, what is the role of the mitochondria? If you just put respiration there, then often you don't get the mark, because in that case, you need to say specifically aerobic respiration, because that's what the mitochondria do. Yeah, in this case, just plain old respiration is suitable. And then I'm going to skip this one because we took a little bit of time to get set up, and we're going to go through carbohydrates anyway. So yeah, we're gonna look at the difference between monomers and polymers. We're going to look at how alpha glucose and beta glucose differ, and we're going to look at the structure of cellulose and starch and how that relates to their function. At any point in the session, if you guys have some questions, yeah, please don't be shy. You can ask questions. And yeah, I'm going to be throwing out some questions like we did just now. And yeah, please do feel free to answer. The more you put into the session, the more you'll get out of it. And I can look at your answers and just check that everything looks okay. Cool. These are the spec points. You should all have your own spec anyway. If not, you can download them. And this is something you should be referring back to a lot throughout the year to make sure that you know the topics that are going to be coming up on your exam. Okay, so who can give me a definition for a monomer and a polymer, or either or both? These are two very important concepts that come up again and again in the biomolecules topic. So we need to make sure we understand these. There's not like one specific definition. It's more like an idea that you're getting across. So yeah, let's see what you guys can come up with. Yeah, I like those. So polymer is made up of a chain of monomers. Nice. Multiple monomers joined. Let's make a really long definition. Joined via chemical bonding. And that now I kind of regret this now. Now the sentence sounds a bit weird. Joined by a chemical bonding in a chain. That's not a great sentence anymore. This is the optional part, and maybe I actually wouldn't put that in because now it sounds a bit weird. But yeah, they are chemically bonded together. And yeah, so a monomer, you say something like a small repeating unit. Can be joined together to form a monomer. Yeah, nice. So these come up a lot throughout the topic. Ones we look at are amino acids and proteins, nucleotides. And the ones we're going to look at today are monosaccharides. And again, we have polysaccharides, we also have polypeptides in proteins, and we have polynucleotides, which are genetic information. We usually just refer to these by the two types, which are DNA and RNA. But the word polynucleotide is one that you can use. Nice. Oh yeah, I forgot the top bit of the screen you can't see, can you, because of the video. I will try not to write anything in there. So yeah, let's go through these definitions. So we're working our way up. So the smallest carbohydrate unit is known as a monosaccharide. We need to know 3 examples of this: glucose, fructose, and galactose. there are also other ones that come up throughout the course of the A level, like for example, ribose, deoxyribose, triose phosphate, pyruvate, GP, RUBP actually is also one. So, but these are monosaccharides that come up in other topics. For example, a bunch of them come up in respiration and photosynthesis. But the three that you need to know when you actually talk about this topic are these three. And they can be bonded together to form disaccharides. So a disaccharide is just two monomers joined together with glycosidic bonds. So let's take one example. If this is glucose, and we join it with another glucose, that gives us a disaccharide. And in this case, this would be called maltose. Again, you need to know three disaccharides, and maltose is one of those disaccharides that you need to know. If you then join a bunch of maltoses together, or just keep adding glucose, you get a polysaccharide, which in this case would most likely be starch. Although I guess it could also be glycogen, but this one's unbranched, so let's go for starch. And we've kind of said already, so polysaccharide is just lots of monomers joined together. Technically, it's just three or more, but in reality, there can be like thousands or even tens of thousands of molecules all joined together. So it's usually a lot more than three. So yeah, let's look at how that happens. So yeah, disaccharides and polysaccharides, we'll be more specific here, are formed by condensation reactions. So a condensation reaction, in fact, let's throw that as a question. So who can tell me what a condensation reaction is? What's the definition? It's actually a very important one. Water. Yeah, it's to do with water. Yeah. So removal of water to produce two monomers. Oh, we didn't produce two monomers though. Lightning Oz. I think you've got everything in your answer there. So you're removing water and you're forming a chemical bond. You need, or you should really have, the idea of forming a chemical bond in your definition. So removal, oh no, that's actually change the wording. Formation, because the bond is the most important thing. Formation of a chemical bond via the removal of water. And yeah, this is done in the process of polymerization. So in this equation, we would need to have plus water in each one. This is a type of covalent bond. And yeah, there are lots of different covalent bonds. We're not going to go into the details of what a covalent bond is today. If you do chemistry, you'll already be familiar with this anyway. Yeah, there's about 4 or 5, I forget off the top of my head. But yeah, 4 or 5 main different types of bond that come up in this topic. Glycosidic, ester, phosphodiester, peptide. Is that all of them? Anyway, yeah, so this is the one that's between 2 carbohydrates. And what that bond would look like is this. So I'm just going to draw a bit of a glucose molecule here. So, actually, no. Hydrogen and OH. So you might recognize this is just a section of an alpha glucose, and this is also another alpha glucose. H and then OH here. So you can see we've got two OH groups in proximity to each other. So what happens when we have that is we remove water. So if we take water out, we reduce H2O. We take the two hydrogens and we take one of the two oxygens. It doesn't really matter which one it is, but one is removed to make water and one remains. So because one remains, that is actually holding the two glucoses together. So let's draw the whole thing. So this would be carbon here, oxygen, and then we're straight into our other glucose molecule here. So that is a glycosidic bond. And I suppose if you actually zoomed in on this in even more detail, it would be a carbon, which is bonded to two other carbons and a hydrogen, oxygen, and another carbon here. So which has also got the same bonding. C. So a glycosidic bond is essentially like a COC bond. Again, this is our glycosidic bond. Cool. Hopefully you guys are happy with that. And yeah, just catching up with the comments. So does the real water mean that the water is produced in the products? Yeah, exactly. So that's one of the reactions. Sorry, not the reactions, one of the products. Yeah, so the reverse of a condensation reaction is a hydrolysis reaction. And because so many of these take place during normal metabolic normal metabolic reactions, this is one of the reasons that water is such an important molecule. So speaking of hydrolysis, we're going to look at this now. So this is the reverse. So how do you define hydrolysis? Bearing in mind, this is how we define condensation. How could you get that key idea into a hydrolysis definition? I'm really glad you guys can't hear the static because it's seriously annoying. But yeah, I'd much rather it's just me that is getting blasted with this white noise than all of you. Some people go to sleep with white noise on, which I've never understood. I find it, I don't know, I didn't think I'd find it helpful. I'd find it distracting, if anything. Yeah, nice. Yeah, some dramatic wording in here. Addition of water to destroy a chemical bond. I like the dramatics of it. Probably I would just say well, I think you'll get away with it in an exam, but it does just sound more dramatic. So I'd probably just go for break though. So yeah, breaking of a chemical bond by addition of water. Yeah, and Christopher Lee, you've said a covalent bond. Yeah, you could specify that. Because yeah, the bonds that are usually broken in this way are covalent bonds. So this is basically the opposite of what we just drew. So I'm just going to draw it very quickly. Oxygen here, and we add in our water, H2O, and we end up with, wow, this is drawn terribly. Should have drawn this hydrogen on here actually already. Otherwise you'd be like, where did the extra hydrogen come from? C. you know, I should probably either have drawn this slower and better or not at all, because this is basically illegible. But yeah, it's literally just the opposite of what we did on this one. So yeah, condensation reactions, hydrolysis reactions tend to be reversible. You can go either way. Nice. So yeah, hopefully so far so good. Feel free to give me a 1, 2, 3 or a sad, neutral, or scared smiley face to let me know how you're finding it. Is that a scared face? Shocked face, whatever that is. And yeah, let's try some past paper questions. Cool. Seems like you guys are all good with that. Well, yeah, I think everybody. Nice. So yeah, let's have a go at these. They're not too hard. Just short, 1 answer, 1 mark questions. Yeah. We do these sessions for 1 hour. Is that what you mean? And yeah, good. If it's not that challenging so far, that's good. It means you know the topic quite well. And yeah, I would say I don't think biomolecules is a particularly hard bit of the spec. Yeah, so it shouldn't be too tricky. So yeah, what have we got this first one? Feel free to chuck in answers out of sequence. So let's call it, well, this one's A. Let's just say B and C to make it easy. So who's got answers for these 3 questions? We probably, on YouTube, we won't get through every single topic in biology. At least like not in a live stream. We have got lessons on every topic on YouTube, but they're not going to be, we only do 1 live stream a month, so we won't be able to get through all of them. But on our website, we do, yeah, we get through the whole syllabus throughout the year, because we do 4 sessions a week, so we progress a lot quicker. Yeah, nice. So we said small molecules. Then we form polymers. Would be a good answer. This is a glycosidic bond. Nice. And let's put one for A. Yeah. So I would say either monosaccharide or glucose. Or if you want to be even more specific, what type of glucose is that? And how do you know? And if you don't know, don't worry too much. Rabia, could guess. It was 50-50, but that's actually beta glucose. And the way that you know that is that beta glucose, oh, some of you guys have said that. I'm just like, yeah, refreshing as often as I can. Beta glucose, each glucose molecule is flipped 180 degrees. So you see this one is pointing up, this one's down, this one's up. The next one will be down, up, down, up. That tells us it's beta glucose, but you didn't need to put that to get the answer. So this is most likely a small section of cellulose, which is a polysaccharide made of beta glucose, which is used in plant cell walls. Okay, so we just mentioned this. Alpha glucose looks like this. The hydroxyl groups are facing down on both carbon 1 and carbon 4. Let's number all of the carbons. So you start at the oxygen in the ring, and you go around. So carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, and carbon 6. And then again, carbon 1, 2, 3, 4, 5, and 6. So on beta glucose, the OH groups on carbons 1 and 4 point in opposite directions. So carbon 1 points up, carbon 4 points down. It may not seem like a huge difference, but actually this is what gives rise to the different properties when they form polysaccharides. So like if you polymerize alpha glucose, you can either get starch or glycogen, depending if that's in a plant or an animal, whereas this one, you get cellulose, which are fundamentally quite different molecules. These are energy storage. Because the glucose, the alpha glucose is used for respiration, whereas this is a structural molecule. And it's kind of like a building material. That's what structural means. And it's used to give strength to plant cell walls. It's basically the main building block of a cell wall. So yeah, they're pretty tough. Cellulose is actually basically what we call fiber is essentially just cellulose. We can't digest cellulose as humans. So we can't access the glucose to make energy, but it does play an important role to just kind of keep everything moving through the digestive system, provide some sort of I don't know what the word is, like roughage or something to keep everything moving through. So it is actually important. A diet deficient in fiber leads to increases in bowel cancer. So yeah, get your 5 a day in essentially. Although they've moved it up now, haven't they? It's like 5 a day is not enough apparently. we should be getting, I think it's like close to 10. So yeah, hopefully that's still so far so good. Then let's look at the two molecules in a bit more detail. So I just refreshed the chat. So I see some questions. Why are beta glucose stronger than alpha glucose? Yeah, good question. It's not that the bonds are stronger. It's just that the molecule, because they're flipped. We're going to cover this in a second anyway, but basically it's because the strands can sit close together and they can form hydrogen bonds, which we're going to look at in a second. What I've just realized that we haven't done is talk about the disaccharides. So I'm going to do this on a blank screen. So we're going to make a table. Monomers, or no, actually change of plan. Monosaccharides. Let's just put that. Disac. And let's fill in this table. So 1, 2, 3. These are the 3 you need to know. I'll do a mixture. So this one, let's say glucose plus glucose. And so this will be answer A. Answer B will be what makes up sucrose, and then answer C will be what makes up lactose. So what does A, B, and C equal? Because you need to know all 3 of these, again, for your exam. Nice. Maltose, glucose, and fructose, very good. and C is glucose plus galactose. Nice. So yeah, if you haven't memorized those yet, just try and find a way to commit them to memory. Do look, cover, what's that? What's it called? Look, cover, write, check, repeat, whatever that process is. Or use flashcards, or yeah, get a family member to quiz you until it sticks. Because yeah, they do actually come up relatively often in the exam. It'll only be worth 1 mark, but it's quite an easy mark. So you don't want to be dropping easy marks, because it's going to be harder to get a good grade in the end. Like, exams are kind of funny. Like, the grade boundaries change each year. They're fairly fluid. So all you have to do, I mean, it's not easy, but to do well in an exam, you need to do better than everyone else. And they sort of put people into banding. So the top 10 to 15%, they put as an A star, and then the next 10% they put as an A, and the next 10% as a B. So really, you're not aiming for a certain percentage. You're aiming to be in a certain percentage of the people doing exams. So you need to do better than the other people. And if you're dropping easy marks that most people are going to get, you make your life much more difficult. You then need to gain back those marks on more challenging questions. So yeah, a very long-winded way of saying, just make sure you're not dropping easy marks on an exam, because yeah, it makes your life much harder. So let's go back to this and let's look at the polysaccharides. So we said the function of starch is energy storage. And this is used to store glucose mostly for aerobic respiration. Then just seeing what we've got on this. Okay, so starch. Some specs talk about amylose and amylopectin. If you're AQA, I think everyone else does other than AQA. If you're AQA, you don't need to know these terms, but what you'll call it is unbranched and branched starch. But the technical names are amylose and amylopectin, and obviously amylose is unbranched and starch. Amylopectin is branched, hence those names. They're only made up of alpha glucose. So like many formed joined together by glycosidic bonds. In both, you have what's called 1,4 glycosidic bonds. Which, if we go back to this diagram, they're between carbon 1 and carbon 4. So these two are going to interact and form the glycosidic bond. If you get lots of 1,4 bonding all joining together, you basically get a long chain, an unbranching chain of alpha glucose molecules, like this. If you chuck a 1,6 bond into the mix, this adds a branch. So these are all 1,4 bonds, 1,4, 1,4, 1,4. If you chuck in a 1,6, you get a branch. So this would be a 1,6, 1,6 bond, and then again you can extend this chain. These would also be 1,4 bonds. Oh, I'm going into the video screen, aren't I? Yeah, maybe let's do it here. So this would be a branch. So the branch, this is the 1,6, and then these are back to being 1,4 glycosidic bonds. So when you have a chain of these, a chain of the 1,4s, they're slightly curved. So what you tend to find is you get this helical structure. The reason for that is to do with the carbon 6 molecule. So the carbon 6 element. So the carbon 6 is on the same side for all of them. when it's alpha glucose, and they slightly repel each other. So if you've got them all on the same side and they're slightly repelling each other, that's going to form a bend. So this forms like this. If you get enough of them, you eventually form a spiral, which we see on the diagram. Nice. Brush this. Starch is just in plants. Yeah. Animals digest starch and turn it into glucose. And if they want to store it, they store it as glycogen. So yeah, glycogen is the animal version of starch. Structurally, glycogen looks more like a amylopectin than amylose because it has branching. In fact, it's even more branched than a amylopectin. I think we don't actually have a slide on that for some reason. Let's go straight to cellulose. Yeah, so glycogen, a more branched version of amylopectin found in animals. Cool. So these are three of the important properties of starch. Why are they all important? Again, you don't have to do these in order. You can just jump in. So why is it important that it's compact? Why is it important that it's insoluble? And why is that one important as well? And yeah, sorry, the second half of your question. Amylase, yeah, amylase breaks down starch into maltose. So starch, maltose, glucose. And the enzymes are amylase and maltase. Let's do that. Yes. Nice. Does not affect the water potential. Hopefully you guys are familiar with this. This looks like a trident. It's actually the Greek letter psi. But it's used to mean water potential. Don't know why they chose that letter. But yeah, so that means water potential of the cell. Because if you think about it, if you just pumped a whole bunch of glucose into a cell, that's going to lower the water potential. Therefore, water is going to move in by osmosis. The cell is going to swell up. Plant cells don't burst because of the cell wall, but it's still going to interfere with the osmotic concentration. So we don't want to do that. So we turn it into an insoluble molecule that has no effect on the water potential. And then, yeah, compact just like helps with storage. Yeah, obviously the more compact something is, the more of it you can get in a smaller space. This is an advantage. It's less important for plants because they don't really move, so they can just store this in the stem or in the roots. More of a thing for animals, but still, you might as well be efficient with your storage. And yeah, this means it can be readily respired. And yeah, this one kind of ties in more with glycogen. So glycogen is more branched. So glycogen has a lot of 1,6 bonding. And the idea behind that is to make it very quick to hydrolyze and break it down. So you can very quickly release the glucose and therefore you can use it for metabolic processes. animals have higher metabolic rates. They need energy in like bursts, and that's why it's so useful for them. Okay, then cellulose we said is cell wall. Gives strength to cell walls. Its structure is beta glucose. Again, it's got 1,4 glycosidic bonds. And it forms straight chains. This is because the carbon 6s are pointing up and down. Each one is pointing a different direction. And the same here. So they form straight chains. These straight chains can then form hydrogen bonds between them. So you often represent hydrogen bonds with dashed or dotted lines. So this is one cellulose strand. And this bundle of cross-linked strands, cross-linked via hydrogen bonds. Bundle of cross-linked strands form a microfibril. And this is what cell walls use to build cell walls. Well, I guess I should specify plant cell walls because there are other types of cell wall. Like bacteria use, you can use either word, murine or peptidoglycan. And fungi use chitin. So yeah, but plants use this molecule, cellulose. So yeah, kind of the same as before. Like this allows to stack and form cross-links. Insoluble, no effect on water potential, same as before. But actually, more importantly, it's a building material. If it dissolved, it wouldn't be very good as a building material. If your house dissolved when it rained, it would be a rubbish house. So you don't want to make something out of something that's soluble in an environment that is very much aqueous. There's a lot of water around. So yeah, we need it to be insoluble. Then hydrogen bonds form between chains by microfibrils. Yeah, so this creates strong group like fibers. Yeah. So again, hopefully that is all good. And yeah, let's try this past paper question. So starch and cellulose are two important polysaccharides. This shows how the starch, how the cellulose. Explain the difference in the structure above. The structure between starch and the cellulose. The 2 marks. So what can you guys come up with for that? So yeah, starch alpha, beta, and put like only, sorry, only 1, 4 bonds, and this one has 1, 4, and 1, 6. Yes, branched. Unbranched. Kind of turn this into a comparison now. Forms H bonds / crosslinks, nice. Someone mentioned the shape. Oh yeah, nice. So helix or spiral, and this is like straight chains. Yeah, nice. That's probably enough to be getting on with. And you can talk, yeah, you can talk a little bit about how they can form microfibrils. Yeah, and they're all the differences. They obviously have similarities, both made of glucose, both have glycosidic bonds, both have 1, 4 glycosidic bonds. But this question was focusing on the differences. Nice, and that's it for today. So we've now completed all 3 objectives. Yeah, if you guys have any questions, I will hang around for a couple of minutes. Otherwise, yeah, thanks for joining. Hopefully see you guys in one of these sessions in the future. Next biology one, I'm actually not exactly sure when it is, but check the YouTube schedule. Yeah, give us a like on YouTube and you'll get notified when these are coming up. And yeah, if you enjoyed the session, do check out our website. Like I said, we have like 4 of these sessions a week that we host on the website, and they're much smaller classes. So you kind of get like a lot more individual attention, so to say. Like, well, I can answer all questions in like detail in our like website sessions because there's fewer people. Yeah. So let's see. Chat. Just refreshing. It's not next week, I don't think. It'll be in a couple of weeks. Check the schedule for more details. And do you recommend using flashcards or a mind map? I think they both have their place. I kind of see what that person is saying. Flashcards are isolated. Mind maps are better at linking everything. But I think it's like horses for courses. Like, yeah, flashcards are just when you want to memorize like definitions or that sort of thing. Whereas mind maps is to help you in your like brain link together the topics. So yeah, they both have a use, but they're for different things. So I would use both. I think flashcards are still like a very good learning resource for things like definitions. But yeah, variety is good in revision. Revision is, as I'm sure you guys are aware, sometimes a bit boring. So if you can mix it up, that makes it substantially less boring. So that's nice. Cool. Yeah, I think just giving it one final refresh. So much memorizing. Yeah. There's like biology is the biggest syllabus, I would say. You need to know the most for biology out of any A level. It's pretty tough. But yeah, that's just part of the package. You've just got to start your revision early, like make sure you're learning stuff throughout your 2 years, because it's just basically impossible to cram it all in the final bit, because there's just so much to learn. The schedule, yeah, I'm not sure, I'm afraid. But I think, in fact, okay, give me 10 seconds. I can look up when the next one is. Oh, it's actually not too long, 2 weeks time. So there's another session on the 13th of December. So yeah, hopefully see you guys there. Cool, right. Yeah, enjoy the rest of your Sunday, and yeah, see you in a couple of weeks if you're coming for that one. Bye.